Hello, my name is Angel Rodriguez, and today I will be presenting the effect of microgravity and space radiation on neuroinflammation and dopamine dysregulation in the mouse brains from Rodent Research 1 mission. Just some quick background. Microgravity and radiation exposure during spaceflight can pose a lot of health risks to astronauts, including the disruption of physiological homeostasis. This can lead to a lot of neurodegenerative neurological risks, more specifically neuroinflammation, which is usually characterized by inflammasome activation. Inflammasomes themselves are just protein complexes that are shown to be activated upon the innate immune response. This process is critical to the immune response following cellular damage. Usually, when you're in space, a lot of the microgravity and space radiation can cause a lot of the cells to just die due to oxidative stress, and that in turn activates this immune response. Um, Additionally, a lot of reduced movement and neuromuscular activity in the microgravity has been linked to alterations in dopamine levels, as well as other neurotransmitters such as serotonin, norepinephrine, epinephrine, which can lead to dysregulation in motor control and cognitive function. The main relationship between inflammasome signaling and dopaminergic dysregulation in spaceflight conditions remain relatively unknown. So the main question that this project focuses on is what does spaceflight affect, or I'm sorry, how does spaceflight affect the brain of mice upon returning? Um, so for this project, we do focus on three different regions of the brain. The first two being the motor cortex and the hippocampus. The motor cortex is mainly found superior to the mice brain and involved in a lot of voluntary movement and coordination. So just how we move from our day-to-day lives. The hippocampus is located posterior to the motor cortex and is involved in spatial memory and awareness. We focus on these two because a lot of the cognitive and spatial awareness is coordinated between these two, and we hypothesize that these two regions of the brain are mostly affected. The third region is what we will refer to as remaining tissue. It's a non-localized tissue, so it's not very specific in function. It's just whatever tissue we had left. So we do have four groups in our sample. We have our baseline group, which acts as a control. It was sacrificed prior to the experiment. We have vivarium and ground, which both were in Earth throughout the whole entire experiment. The only difference is ground experienced the International Space Station similar conditions, so similar to our flight group, which actually went into the ISS for 37 days and then brought back down. So I first would like to talk about some activity scores between our flight group and our ground control group. So again, this is between the group that experienced actual microgravity and spaceflight conditions versus our ground control, which experienced simulated conditions. As you can see, our flight group is labeled blue and our ground control is labeled red. Initially, in these first few days, you can see that our ground control group actually was more active in the activity score compared to our flight group. But around day 11 is when we see this jump from very little activity to the max amount of activity that is scored from our flight group, as well as our ground control group sort of mellowing out. A lot of this can be due to sort of adapting to the microgravity. There's a YouTube video that kind of explains this really well, where the mice actually adapt them to the microgravity and start kind of using the microgravity to their advantage for movement. So it ends up becoming more of an enrichment for them, which is what we didn't expect, actually. We expected more of the first few days to kind of show what was going to happen during our experiment. Next, I want to look at the dopamine analysis from remaining brain tissue. Again, it's the non-localized tissue. So it's just whatever tissue we were able to collect afterwards. A lot of the times we didn't see any changes or significant changes. The main things we did find were norepinephrine, which showed a higher concentration of norepinephrine in our vivarium samples, so just our ground samples. Between our baseline, our ground, and our flight, we didn't see much of a change. However, there was significance between our ground control and our flight samples. Again, it's shown in dopamine, which there was a change between our ground control and our flight samples again. That shows that there's just some changes between the metabolite concentration between these two groups, which can simply put, that there could be changes between simulated microgravity compared to the real thing. Next, we did experiment on microRNA analysis. So first we have a heat map. First, on our left side for the motor cortex, we have our ground control, and our right side, we have our flight control. The blue is to indicate an inhibition or a downregulation, while the brighter orange and red colors are to signal upregulation or activation. We are mainly able to see that for our flight control, a lot of major genes are actually activated compared to our ground control, which are actually inhibited. Looking down to our Enrich Go map of the microRNA functions that are affected, a lot of them are based on membrane transport between ions. So a lot of it being potassium, chloride, monoatomic anions, and protein local and response to vitamin D as well. Um, One thing we were able to look at is between these ion transport membranes, a lot of them are in turn related with inflammasome activation, mainly potassium. So being able to see this kind of does prove that there are some changes to inflammasome activation or some changes between how the ions help move this inflammasome activation. Moving to the hippocampus, on our left this time we have the vivarium labeled in red and our ground control labeled in blue. Majority, we can see that vivarium shows a lot of downregulation and ground control shows a lot of upregulation, but the top genes that were shown to be most affected shows the opposite. So our vivarium genes had upregulation while our ground control genes showed a downregulation. Just to go back to the EnrichGo mapping of the microRNA in the hippocampus, we did see that a lot of the changes were mainly microRNA that are involved in ensheepment of neurons, as well as myelination and oligodendrocyte differentiation. What this can mean is the hippocampus, mainly involved with spatial memory and memory in general, does involve a lot of pathway differentiation as well as just forming new pathways. So a lot of this myelination changes can be inferred that a lot of the changes that happen during spaceflight when it comes to changing muscle memory and sort of adapting to the new environment caused a lot of the microRNA genes to become activated and express a lot more. Um, our last thing I want to talk about is the gene expression of general inflammatory genes as well as expressions. These were based off the remaining tissue, so again, not localized. We didn't see much significance between DJ1, NLRP3, caspase-1, or IL-1 beta, which are the main inflammasome genes that I wanted to focus on. So I did go ahead and focus on some major cytokine genes as well, which is IL-10, TNF alpha, and IFN gamma. What we did see is that there are some changes when it comes to expression. An example could be the TNF alpha, which shows vivarium in flight showing an upregulation. However, it doesn't show a lot of significance. So although it does show expression, it's not really something that we can sort of um, claim that it is significant. So what we've seen with all this is although gene expression through RNA and PCR shows that there's not a lot of expression, not a lot of significant expression changes, we did see a lot of microRNA changes within the cells. And yes, thank you so much for this presentation.